Okay, so this is a talk which is a paper that just came out in Philosophy of Science, and is part of a broader project that I've been interested in the philosophy of neuroscience and thinking about the ways in which optogenetics influences the way that we study and theorize about the nature of memory, particularly in the case of the neurobiology of memory, how we're thinking about it at that level. So the real centerpiece of the paper is to talk about optogenetics, which I assume is a term that at least is familiar with most people. If you aren't aware of the details of what optogenetics is, I'll be saying more about that later. But the basic features of optogenetics is that it's a new intervention technique that allows for the manipulation and control of neurons via making them light responsive. So neurons can be turned on and off, more or less, by exposing them to light. It's a way, in a way, it makes good on a promise that, or a hypothesis is too strong of a word, but a speculation that Francis Crick made quite a while back, that one of the new frontier for intervening in and making progress in neuroscience would come from finding some form of light manipulation in neurons. And the sort of making good on that promise or speculation came from Carl Diceroth, who's pictured here, who's at Stanford University. It was him and his team who really engineered the opsins and got optogenetics going. The first paper was in 2005, so it's not even been around for 15 years yet, but it's already exploded as a technique. It's been declared a breakthrough of the decade, method of the year. The accolades are constantly coming, and presumably the Nobel Prize is not far behind. So the technique gets a lot of hype and attention from scientists, neuroscientists in particular, but it's starting to get attention from philosophers as well. And this paper in particular was in part put together initially as part of a symposium of people thinking about whether or not optogenetics is actually worth all this hype. So here are just a few instances of the kind of hype that optogenetics has been getting, right? We talked about it ushering in new eras of control, that it's allowing neuroscientists to direct symphonies of light-induced neural activity. I really like that mental image. People asking me sort of big questions about whether optogenetics is actually going to revolutionize the way that we have access to and control over our behavior. The possibility of intervening into disease and mental disorder, and that this might, as some are speculating, fundamentally overhaul the way we think about ourselves as human beings. So these are the kinds of things that scientists and science writers are frequently saying about optogenetics, and it's a real question for philosophers of neuroscience, whether or not it actually leads up to the hype. And so in thinking about optogenetics in this paper, I'm really thinking about it as an intersection of two questions and concerns in different parts of philosophy of neuroscience. The first is a set of concerns about tool development and the role that plays in revolutions or developments and advances in science, and Engram theory and the science of memory, and how the use of optogenetics actually allows us to make progress there. So I'll say something about each of those really quickly. So on the question of tool development and scientific revolution, the point that's being made here very frequently by both John Bickel and Carl Craver is a sort of way of making a more specific application of a broader, more general point that comes from Ian Hacking, that much progress and development and revolution, if there is any in science, comes not from theoretical developments and advances, but actually from new abilities to do stuff, the ability to make new tools, to have new kinds of interventions. Once we have something like this to hand, we can actually do new stuff. And so for philosophers of neuroscience wanting to apply that point in particular, optogenetics is quite exciting because it looks like a scientific revolution occurring in real time. So it looks like we're watching a new tool develop and be used to change the way we think about different fields of study. And so there are questions about whether optogenetics really is kind of playing that role. And of course, one of the best ways to think about whether it's playing that role is to look at particular areas of inquiry. And so one that I'm interested in more broadly is what you might think of as Engram theory and the role that it plays in memory science, particularly in the neurobiology of memory. So it seems that in many areas of philosophy and in at least most parts or some parts of memory science, we're convinced that there is some sort of engram or memory trace. I'll say a little bit more about what that is in a minute. So why is it that we're so convinced that it exists? What are the sort of arguments that support that kind of a claim? What is the engram supposed to be? What kind of mechanism is it? Where exactly in the brain are these engrams? How is it that they work? And one concern that's become more pressing for many people in the last say, decade or so, is whether or not this kind of commitment to the idea that there is going to be an engram is compatible with memory errors. So part of what this paper is really doing is asking these questions about the ways in which optogenetics has been revolutionary in neuroscience by looking at the ways in which optogenetics has allowed us to intervene in and do things to the engram that have had an influence on engram theory and the ways that we think about memory in its neurobiological study. So this paper is all about, in particular, optogenetic intervention into the engram. So here's a set of claims that I'm going to make today. And I think that, especially when I wrote this paper, so I first started working on this stuff a few years ago, and going back to this now, and in light of where my thinking about optogenetics and memory is now, I realized I was in a moment of serious optimism and deep faith and excitement about optogenetics when I was writing these things. That is, as you might infer from my tone later on, starting to wane, at least in small bits, or I'm starting to think it's a lot more complicated. But I do think that optogenetics has been revolutionary for the study of memory, particularly for the ways it allows us to intervene into and manipulate the memory processes at a lower mechanistic level than we've previously been able to do. And I think that the technique solves a key methodological challenge for investigating memory, a challenge I'll introduce in just a little bit, and that's the challenge of separating the engram and the Esphory. And I also think the use of optogenetics, because it allows us to defeat that methodological challenge, has produced some significant advances in how we think about particular forms of explanation for certain kinds of memory errors that we see. So it has done all of these things. Those are the claims that I make in the paper. The point that I have listed here is claim 4, is the sort of my developing curmudgeon about some of these things. I don't think that optogenetics is a panacea. I don't think it's going to answer all of the questions that we have about the nature of memory. And I think it actually, the further it goes, the more it introduces a new set of conceptual challenges for how we think about the study of memory. So it hasn't, it's not going to solve everything for us, although it is making for some exciting advances. So here's what I'm going to do today. I'm going to say a little bit about what Engram theory is. Then I'm going to introduce this idea of what I'm calling a methodological challenge for how we investigate and intervene into engrams. I'll then give you my very fast non-scientist introduction to how optogenetics works, and then talk about the ways in which optogenetic intervention has been used to tag and manipulate engrams, particularly the work out of the Tanagawa lab at MIT. And then if there's time, I'll say something about a few challenges or objections to the line of thought that's been developed in this work out of the Tanagawa lab, and my sort of glowing claims about where the field could be going. Okay. So first, Engram theory. So when I say engram, I guess as the first pass of telling you what I'm not talking about, I'm not talking about engrams as the concept or term is used in Scientology, if any of you are familiar with it from that particular form of usage. I'm talking about engram as what we might think of as a newer, at least it's only a little bit more than a century old, a newer term for a much older idea, which is the idea of a memory trace, or the idea that in the life of us or other organisms, as we go about things in our world, we have experiences. Occasionally, those experiences make some kind of impression on us. They induce some sort of change in us, as a result of which we're then later able to think about those experiences. And whatever that change is, whatever that sort of marker signature of the previous event, experience, or information is that's left behind that we later can then use to recall the event, that thing is the memory trace or the engram. So at different points in time, people have thought about memory traces as impressions in wax, as images or items in a warehouse. as grooves in phonographic records. And the engram is a sort of attempt to take that general idea and commitment and direct it in particular to thinking about what the neural mechanisms or brain mechanisms of that memory trace process might be. The particular term engram, as it's used in the neurobiology of memory, comes from the German biologist Richard Semon, who was writing in the early 20th century and coined the term engram. You can sort of I think of Semon as the sort of Mendel of memory. So in the same way that Mendel had this idea that there would be genes, there's a sort of commitment he had to the idea that there was going to be this thing that was explaining heredity and reproduction, but he sort of postulated that before actually having made the discovery. Semon did the same thing for the idea of an engram. There must be something, some mechanism, some sort of physical or chemical structural change that occurs as a result of learning and which is reactivated during remembering, but he had no idea what it was. He didn't have much in the way of a direct hypothesis about what those mechanisms would be, just that there needed to be such a thing. And so there's a way in which that sort of promise has been carried forward into what really developed into the neurobiology of memory, which is centered around the study of engrams. But most people, Semon's becoming more sort of coming back into people's awareness more recently. But when most people think about engrams, they often think about Lashley, Carl Lashley's somewhat famous search for the engram. So Lashley did a series of studies over the 1930s and 40s and early 50s looking for the engram and quite famously failing to find it. So doing studies in rabbits and other rodents where he would lesion parts of the brain and see whether or not he could locate the particular structure responsible for learning and memory, and he was never successful in doing this. So he wrote extensively about his belief that there was no such thing as an engram, that instead the brain was going to be sort of equipotent in its ability to structure and store information. But whenever I talk about Lashley, I always like to fall back on this quotation from him that I'm showing here, which is that even for somebody who spent most of his career railing against the possibility of the engram, the idea that there's going to have to be one in order to explain what happens in instances of learning and memory is sort of foundational. So one person's modus ponens is another person's modus tollens. So if you're committed to the idea that there has to be one in order for learning and memory to be possible, Lashley sometimes feels himself compelled to reject the idea that learning is possible, given that he can't find the engram. And then here is sort of the history of the neurobiology of memory in one slide, which is just sort of generically pointing at this idea that after Lashley's concerns and skepticism, we got over them through a series of discoveries. I'm just going through this very, very quickly here, but the idea of how we might see particular interneuronal communication in ways that might suggest a mechanism by which, if two neurons are in communication, that communication can get stronger and stronger as a result of prior experience. What we now, thanks to Lomo and others, think of as long-term potentiation. That has looked to many like a very good mechanism for being the sort of basis of potentially of engrams in learning and memory. And through the work of Brenda Milner and others with patients like Henry Meliasen, who we knew for most of his life as HM, we know that the hippocampus and its surrounding structures are critical to the process of remembering. So the idea of the engram remained the sort of guiding conception for much of the work that's done in the neurobiology of memory, and now these kinds of broad concepts frame a great deal of the investigation that happens into the nature of memory at this level. And so I just put this particular quote here from the paper out of the Tanagawa lab from a few years ago, talking about what Engram theory looks like now, where they make explicit reference to Semon's initial view, and sort of what they've done is really not much of an advance on what Semon himself had to say, right? Just saying that when a subject undergoes an episode, there'll be a set of selected stimuli from the experience, so we'll activate a population of neurons. It's a slightly more detailed articulation, but it's still sort of functional and generic about the details of the mechanisms. But they take this to be the basis of what they call Engram theory, which is a sort of formative way of thinking about how to investigate the mechanisms of memory in neurobiology. Right, so that's my fast and furious version of Engram theory. So now I want to talk a little bit about what I take to be a methodological challenge for the success of Engram theory. In part, the challenge is in the title of the view itself. So in Engram theory, the focus is on the engram. A recent Nature Reviews Neuroscience paper from Jocelyn makes this clear. So there's a lot of talk about the ways in which the development of new tools is helping us make progress in finding and discovering and manipulating the engram with a real focus on that particular construct and mechanism. But if we return to what Semon actually had to say originally, Semon didn't just posit the existence of an engram. Semon posited that there was both an engram and an Esphory. So this is a picture. I don't expect the picture to make a great deal of sense to you, but it's from Semon's book, The Meme. It's very hard. I never know how to say that word from him. But the idea was that remembering requires both the engram and an exphoric process by which that engram is re-excited in the process of retrieval. So many people think there's a fairly straightforward translation between Semon's use of the word engram to memory trace and Semon's use of the word Esphory to retrieval. In fact, Semon went so far as to have both a law of engraphy and a law of Esphory, basically saying everything that we're capable of learning and remembering is going to be laid down as some sort of physical, chemical, structural changes in the brain, and it will be through a reactivation of these processes exphorically that reinstantiation or remembering can occur. Right? So critical to Semon's view is not just the idea that there would be such an engram, but that exphoric processes would be involved in the retrieval of that engram. And I think that point, insofar as Semon is talked about at all, he's talked about as the father of the term engram, but not these two terms in conjunction, particularly not in lower level studies of memory. And in higher level studies of memory in cognitive neuroscience and cognitive psychology, scientists know well over work over the past several decades that retrieval is not a neutral process. So the process by which one activates information to be retrieved in memory is not some sort of neutral probe that does not itself interfere in the act of remembering. And so it's important to keep an eye on that process and think about it as playing a role in, or as potentially having an influence on what happens to the engram. So we can think about the engram, sort of what is stored, and the Esphory, the process by which the thing that is stored is retrieved, as two distinct things. Those are distinct entities and processes. But the challenge comes, the real methodological challenge comes from the fact that we never get to access the engram by itself. The engram is always accessed via the Esphory. So we only, I mean, you can think about this in your own life. Sometimes you just don't know what it is you actually remember until you're put into a condition where, for some reason or another, you need to retrieve it. This is something I'm always reminded of when you're on a long car trip and you're sort of scanning through the radio and some song comes on you haven't heard in 20 years and all of the lyrics come flooding back to you, and you realize that you had no idea that that was in there until something produces the circumstances that help to bring it to mind. And so there really is no way of accessing the engram without the Esphory. And so we want to be able to distinguish the role of those two processes. Now, when we're talking about explaining cases of successful remembering, where someone, intends to retrieve something and that information, the engram related to that is retrieved and the information comes out, we don't really worry so much about the role that each of those processes are playing because we assume that they're complementary and everything is working as it should. But the methodological challenge really becomes a challenge when we think about cases of memory error. So when we want to try to figure out when there was an error, when something went wrong in the memory process, was the challenge a failure of the engram or a failure of the Esphory or something about the interaction between the two of them? So to put the point very briefly, or generically, we can think about there being broadly two different kinds of memory errors. There can be errors of commission and errors of omission. So false memories, where we produce memories that are either partially or wholly false about a past situation. So, remembering that you ate a salad for dinner last night when in fact you had pasta, that would be an instance of false memory. Whereas if I ran into you this morning and I asked you what you had for dinner last night and you just couldn't tell me anything, that would be an error of omission, an error of forgetting. And in each of these cases, we can worry about or wonder about whether the failure is a result of the engram or the Esphory. So in cases of false memory, is it the case that I produced the memory of eating a salad because there was no engram left there, and I just produced something that seemed likely or plausible given that it was a Thursday night? Or is it that the engram was there, but there's something about the way that the question was asked, or the particular circumstances that I'm in, or the particular nature of that Esphory process that distorted the memory that is there and led to the error in question? In cases of forgetting, right, if I failed to produce the information, you ask me and I just cannot call to mind what I had for dinner last night, then it could be because that information simply isn't in there. I don't have an engram corresponding to my meal, or it could be that that information is in there, but I just lack the ability to retrieve it. I lack the right kind of process that would allow me to access it. And so when we want to know the difference between those two, if we don't have a way of disentangling the influence of the engram and the Esphory, we're stuck without a way of disentangling those and answering those questions, and so really giving a thorough explanation of cases of memory error. Okay, so that's the challenge that complicates the study of memory. And now I want to talk about optogenetics, in part because, as I revealed before, this is a very exciting way for potentially getting around this methodological challenge and giving us new insight into these questions, in particular about memory errors. So optogenetics is an intervention technique by which living, acting, in real-time cells can be made light responsive. So it combines practices that have been standard for several generations now involving genetic modification and engineering in model organisms with a particular kind of protein. These proteins are opsins. They're light-sensitive proteins. So many of them come from algae. There are some now that I think are being artificially created. But it's the ability to make these part of what can be genetically modified in model organisms that makes optogenetics possible. So here is my very fast and furious way of trying to explain to those of you who are interested how optogenetics works. So here's a picture of 4 different microbial opsins. This comes from a paper out of the Diceros lab. Channel rhodopsin 2 is the one I'm going to focus on later because this is the opsin that's used in the Tanagawa papers that I'm going to talk about. But each of these particular proteins is such that it can be activated by lights of particular frequencies. So channel rhodopsin 2 responds to yellow light, others respond to blue and green, etc. The way that the modification works is that you find a way of packaging the opsin DNA into a virus vector that can then be given to your model organism. So without going into all the details, in part because I'm sure I don't know all of them well enough, the way that the microbial opsin is put into a virus vector to then be given to the model organism involves both using the transgene, so both putting the opsin DNA into the virus vector, but also using engineering a particular binding site for the promoter proteins so that you can selectively target particular neurons. And that's what's part of what's especially exciting about optogenetics is that you can use these proteins which will be light responsive, and you can design the virus vector such that the cells that will receive and then express this light responsive protein will only be those of particular types of neurons in a particular region. So it's effectively going in and intervening and putting a light switch on a very, very particular set of neurons, as I understand it. So once you've made the opsin, once you've got your sort of virus package for the opsin, then you need to give it to the organism. That's either done through some sort of stereotactic surgery, as you see displayed here. I can't bring myself to show you actual photos of real animals. I prefer the sketches. Or there are now actually opsin transgenic mice. You can get mice that have already been genetically modified to have them. And either way, you're sort of allowing the virus to run its course to infect the organism, and then as the virus activates, these proteins will be expressed in the particular regions where the promoter has taken hold, right? So then, once your organism has the virus, has the opsins incorporated into the particular pieces of neural tissue that you were wanting, then you can activate that opsin. So as you see here in this photo, this is usually done by an LED light cannula that's put near the part of the brain where the opsins have been left. And you can see here just a quick table that shows you different, sorry, I said channel rhodopsin 2 was yellow light earlier, I meant that it's blue. But each of them can respond to a different, is sensitive to a different kind of light, and in response to the light will do a different thing. So some activate in response, some inhibit in response, but they are activated very quickly. So this is an instantaneous reaction. If the light shines, the particular the ion channel opens or closes or whatever it is that you need that opsin to do, which is a very, what is fascinating and exciting for neuroscientists about the use of optogenetics. I put up here a little picture of doxycycline because this and other things are often used, and they're used importantly in the studies that I'm going to talk about. So doxycycline is an antibiotic. If you get a sinus infection, you might even be prescribed it. But when mice and rats and other animals are given this through their water or in some other way, this can control transgene expression. So the animal, if the animal is on dox, as they'll say, if the animal is taking the dox, then none of the genes for the light response that contain the light responsive opsins will be expressed. It's a way of sort of keeping the virus activation in check. Yeah. I just heard something. Am I okay?
Yes.
Okay. So this is a way of, so you add in the fact that now you can activate, you've got sort of light tag on a particular set of neurons, and that gives you very, very quick sort of temporal and spatial resolution, and then you can add a further level of control by keeping the organism on doxycycline at particular times so that the possibility of certain cells being light tagged only happens during the times in which you want it to. So hopefully from this brief and very sketchy presentation, you can see what's exciting to neuroscientists about optogenetics, why many people think of this as a revolution and are tempted to say the kinds of things that I was putting on those initial slides. And that's in part because it gives you highly selective and instantaneous intervention into the activity of living and behaving organisms. So you can watch videos as part of Ed Boyden's TED Talk and various other places where, when you have these optogenetically modified organisms, the mouse can be running across the room and you turn the light on and the mouse will stop. It will, it will completely change behavior in whatever way it is that you've set it up to do this in real time. It's a really sort of fascinating level of both selective and quick intervention while the organism is still living and breathing and behaving roughly as normal. So that's, in general, what has everyone excited about the nature of optogenetics. In particular, many people are excited about the possibility of scaling this up to higher organisms, in particular to humans. There are lots of talks about clinical applications in the cases of things like addiction and particular forms of mental disorder. I don't have much to say about whether that's going to be possible or not, but that's part of what people find so intriguing and exciting about this, including Diceroth himself. But in the case of memory, what's particularly exciting about it and what got me, this is really what brought me to the table of thinking about optogenetics, is that in the case of memory, optogenetics makes it possible to reactivate the engram directly and sort of skip or circumvent standard ekphoric processes. So that's what I'm going to say something about now. So I'm talking in particular about a range of results out of the Tanagawa lab. There are other studies broadly in this ballpark, but this is a very, very elegant and nice progression and series of very well done papers exploring how to optogenetically intervene into engrams. They're focusing particularly on spatial memory or contextual conditioning in mice. The engrams that are associated, we know from past work by them and many, many others, that the sort of memory traces, the engrams for spatial memory and contextual conditioning in mice occur in the dentate gyrus, particularly the granule cells, so that part of the hippocampal formation. And the opsin that they're using in particular through most of these studies is channel rhodopsin 2. And part of why optogenetics works so well for studying and manipulating engrams is that the activity involved in encoding a memory. So if I'm going to go through the act of retaining or holding on to information or experiences that are coming at me or being presented to me, in order to do that, the neurons that are involved have to undergo significant modifications, including more transcription and translation and expression, right? So they're going to actually, you're going to get the kind of genetic activity that's needed to get opsins going and get optogenetic markers working. So if the neurons involved in that memory-forming region, this particular part of the hippocampus, have this opsin protein there and waiting, then if an organism forms an engram, then you'll get activity in cells that will then become light responsive. So you'll effectively have created an engram, and given it a light switch all at the same time, if that makes sense. Sorry, I feel like I end up resorting to a lot of metaphors in the ways that I talk about these things, and most of the time the way that I confirm that it makes sense is that the people looking at me don't look at me like I'm crazy, but I'm just looking at myself right now, so I don't know whether or not it's crazy. So I'll just wait on you to tell me that later. Okay, so here are a couple of pictures that show you something of what's going on. So in this top picture, in the top corner, that purple set of balls or bubbles is a schematic depiction of, let's say, neurons in the dentate gyrus, right? Individual neurons, showing you that when neuronal activity happens, if a particular neuron in this area of the mouse brain, again, these are these optogenetically modified mice, if a neuron is activated, then that will involve the expression of the channel rhodopsin 2 protein here. And so those particular cells, the ones that have changed to yellow, are ones effectively that have expressed opsin proteins, meaning that those cells are now light responsive. So they're not necessarily on at that particular time, but they're cells that now, if they're exposed to the light, will become activated. Right? So that's how this works. So we're looking at the particular mouse brain here. We see what happens. You've got a mouse that has the cells ready to do this sort of thing. You put them into a particular conditioning chamber and you shock them, and that will involve the formation of a spatial memory for that environment and what happened there. And then you'll be able to see that, in fact, those cells have the particular label. And so when you shine the light on those cells, when the blue light is exposed, only those cells in the dentate gyrus, the ones that have expressed that opsin, will become activated. So effectively, this is what I've been saying when I say that you've given the engram, the formation of the memory as a result of the context where the mouse was shocked, you've given it a light switch. And then in this final picture, what I've shown you, what I'm trying to show you down here, is part of a schematic that shows you the green box behind or underneath the set of dentate gyrus cells is a reflection of when the animal is on doxycycline. Right? So if you have the animal on doxycycline, so you've got your animal, you let it hang out in the cage for a few weeks, maybe you've got it on doxycycline the whole time, so that while it's having lots of experiences, it's not forming any engrams that are getting this particular light switch. But when you put them, so you've got the animal doing its thing, and when you decide that you want to put it into the conditioning chamber that's particularly related to your experiment, you take them off doxycycline, add that, and time it just right so that now any new engram that's formed during that process, anything will express that protein, will then become light switch activated, and you'll know, because you'll put them back on doxycycline just after, basically that the only engram that has a light switch is the one that you were controlling for. Right? So this is the way in which you selectively create engrams that have this light switch, even though all of the cells in the dentate gyrus have the ability to express these opsins once activated. Only the ones that do it during your experimental conditions actually have it. That's what the doxycycline allows you. Okay, so this is just a way of expressing in more detail, now that I've talked to you about it, why I think this is revolutionary for this methodological challenge for the study of engrams, and that really this ability to artificially activate directly the engram circumvents standard exphoric retrieval processes. So this avoids the methodological challenge in many respects. So it avoids it just in general, in that we don't have to go through any process of retrieval in order to have the engram reactivated. That's especially helpful in the case of working with non-human animals because, maybe I can, instead of waiting around, if I'm trying to do a study on human memory, I don't have to wait around for you to think about a particular memory from your childhood. I can simply ask you about it and get you to retrieve it. But if I'm working with rats or mice, I can't simply ask them to retrieve it. The only way to get them to retrieve something is to put them back into that context. So the ability to optogenetically activate artificially the engram without taking the animal back to that original context gives you a sort of new range of ways of controlling and then manipulating that engram. And that's part of what has made some of the really exciting developments that I want to talk about now for the bit of time that I have remaining. Right, so it allows us both to avoid that challenge and opens up these new possibilities. So the first case I want to talk about are false memory cases, where what I mean are inaccurate representations. Remember, these are the commission errors, where you produce a memory, something that feels like a memory, but it's either partially or entirely false. It's not something that happened to you, or at least it didn't happen to you in the way that you remember it. And these memories are the bread and butter of the cognitive psychology of memory for the last 40 to 50 years, in part due to results of things like the what's known as the DRM effect, the de-Schrödinger-McDermott paradigm. It was just a quick way of showing this to you, which is that if I give you a set of related items, they're often related words, and then I later ask you whether you remember them, if the set of words here are all related to illness, things like nurse, sick, medicine, lawyer. If I ask you if hospital was on the list, you'll get it right. If I ask you if something like apple was on the list, you'll get it wrong. But if I ask you if something like doctor was on the list, many people will say the word was on the list, they remember seeing it, they remember exactly where it was, even though that word was not actually on the list. This is a very robust finding. It's a sort of modern-day Stroop task for many other manipulations in cognitive psychology and neuroscience. But it's just one general way of showing that if you are exposed to a set of information that's all highly related to a particular concept, but not that concept directly, it's a very frequent false memory error for you to think that that thing that wasn't mentioned but is related was part of that experience. So that's a very, very common and easy to instigate false memory. Another long-standing paradigm of keeping track of false memory comes from Elizabeth Loftus, in particular her misinformation paradigm, right, which shows that if you give people videos, you show them a video of a car accident, if you ask them, depending on the kind of question you ask them, you'll get different reports of the rate of speed at which, according to this car, the blue car was going when it hit the red car. So if I say how fast was it going when it contacted the car versus hit, bumped, collided, or smashed, for each of those more severe words, you'll get a higher estimate of the rate of speed of the vehicle, showing that the way in which you remember the information can be influenced by the way in which you're questioned about it. So showing that presenting misinformation to you can distort the memory in particular ways. These are just two results out of which one can, there's many more of these kinds of things to say, but these sorts of results are everywhere in the human study of memory, and in fact are one of the confounding features both for memory scientists and for philosophers of memory, and thinking about what the nature and purpose and function of memory is if these kinds of memory errors are not just possible, but actually frequent and easy to produce. And so we can wonder, what's making them happen? Are these false memories? Is it because there aren't any engrams there, so we're not keeping track of information? Or is it that there are engrams there, but they're just being distorted by the processes of retrieval? And a standard way of thinking about this is to think that the claim actually is that there is no engram, or and often this kind of argument comes from thinking that it's not simply that these errors occur, but that they occur so frequently that it no longer makes sense to think of remembering as aiming to store information, because if it intended to store discrete information about particular events, we'd be forced to think that memory malfunctions most of the time, and that's a non-desirable result. And so instead, we should think of memory as doing something else. And so these kinds of views are known as constructivism and simulationism. Sort of broadly speaking, Felipe has been one of the champions and clearest articulates of what this kind of view can look like. He and I enjoy, I think, trying to figure out, trying to argue with each other about whether or not this is right. It's generally a way of thinking about memory, sort of rethinking the way we think about what the nature and purpose of memory is, by saying that memory is a capacity for generating simulations of how past events might have gone, rather than relying on relying on general networks of information rather than on discrete engrams or pieces of information. That they're really, what memory errors show us is that that's not how memory is organized and designed. And I think there are a lot of nuances to the kinds of views that people are give in this context. I'm not doing them any justice here. But one of the things that's always been perplexing to me is that, first of all, studies of false memories are exclusively studies on humans. They're behavioral studies of what we do in these kinds of cases. And when it comes to having a sort of mechanistic picture of how the act of remembering or misremembering or false memory works, most of our mechanistic accounts of these kinds of cognitive processes come from animal models. But the difficulty is that we haven't had any idea what a model of false memory in a non-human animal would even look like. So that hasn't been, there's been a real disconnect between the way we think about the theorizing and investigation of false memory in human memory, and the way that we think about the mechanisms and explanations and theories of memory in lower level neuroscience. And it really wasn't clear for a long time what you would even do to start to bridge that gap. But via Optogenetics, the Tanagawa lab in 2013 produced what is arguably the first non-human animal model of false memory. So, and here's how they did it. So here you've got the picture of the mouse. The mouse, the green here is indicative of the fact that the mouse is on doxycycline. So, oh, sorry, if I sit still in my office for too long, my lights go out. So clearly I'm not animated enough when I'm just talking to myself. This is a schematic representation of the cells in the dentate gyrus, all of which have that potential opsin to express if they're activated. So the animal's on doxycycline right now, so none of them are being expressed. The mouse is taken to what we'll call context A. It's a particular conditioning chamber. They run around. They get to explore it. They're off doxycycline at that time. Notice it's not green. And so they form an engram for that environment, for that memory. And because those were all genetically modified cells, the engram that's expressed now has expressed those light-responsive proteins. All right, so now the animal's put back on dox, so there's not going to be expression of any more proteins. They're taken to a new context B, and when they're in that context, the blue light shines, and while the blue light is shining, because this is the LED cannula that's in their dentate gyrus, that activates the engram from context A, but while they're in context B, they're shocked. They're given a set of foot shocks. And so now what they have is an engram that was formed here, but has been activated and given new information, distorting information, arguably, in this second context. Now the question is, what do you do? So you made a light responsive engram and you manipulated it. What does the organism with that engram do in different particular contexts? So if you take that organism and you take them, we'll start over here with B and C. If you take them to context B, this is a place where they got a foot shock last time, and you put the animal there, they remember that they've been to B, and they remember that bad stuff happened there, and they show characteristic fear behavior, they cower, they hide in the corner, etc. But it's not as if what you've done is just made them think that, so in B, they'll cower as expected. In context A, they've been to context A before. Remember, when they went to A, they were just exploring and running around. But now when you put them back in context A, they show recollection. They treat it as a familiar environment, that they've been there before. But they show that characteristic fear behavior that they showed in B. So they will cower and shake and fail to move. And that behaving in a way that's not reflective of what actually happened to them last time they were in that environment. But lest you think that what's happened is that you've now just made the mouse terrified of everywhere that they go. If you put them in a novel context C, which is a place that they haven't explored yet, they show characteristic exploratory behavior relative to novel environments. And what this has been taken to show is that what it looks like what you've done is create a false memory for context A. So what you've done is taken, well, I remember that I was there, but I sort of misremembered what happened to me in that environment. I've mislabeled that information in a particular way. In a second study that expands on this graph isn't particularly helpful. It's the same method as before, where you have the initial context and then the second context, and then you're going to test in 3 contexts. Only what's changed in the second study is that you've added valence to the initial context. So in context A, instead of just getting to freely explore, the mouse either is rewarded or is given that fear conditioning. And then in context B, half of the organisms are given consistent activation, and the other half are given the opposite. So if they explore and form an engram in context A that results in foot shocks, then when that engram, they're taken over to context B and that engram is activated, now they're given pleasant experiences, right? And the reverse. So if it was originally pleasant, now they're being given foot shocks. And the real question is, what happens to that organism? So I've got, originally I was looking around in context A and it was bad news there. I got foot shocks. And then I went over to context B. That engram was activated, but then really great things happen. Usually this is the ability to interact with the mouse of the other sex. That happens to me now in context B. The question is, what will happen to me, but during that time the engram from context A was activated, what's going to happen when I go back to context A? And what they show is that the kind of false memories that you create are that the mouse now has when they go back to context A, they think that good things happen to them there. So they treat it as a familiar environment, but they take the positive conditioning to be reflective, or the positive experience to be reflective of what happens in that environment, not what actually happened to them in that environment, which was getting foot shocks, or vice versa, in cases where the valence is reversed. So these studies are first just cool because they provide really a first demonstration of what false memory in non-human animals could look like and start to sketch a possible mechanism for false memory formation. I also think, and this is a point I've argued elsewhere, that there's a way in which these kinds of results challenge one of the standard constructivist and simulationist approaches to explaining these kinds of false memories, which is to say these errors really suggest that we should reject the idea that there ever are engrams. Instead, it should be these more generalized processes that are forming these memories that become distorted. Instead, these kinds of studies show you that what might be happening in at least some cases is that there are engrams, but it's something about the reactivation process that can distort a memory in a particular way and then produce a false memory. So there are ways of, this gives us at least the beginning sketches of what could be a mechanistic model of explaining false memory that keeps a central role for engrams in that explanation. Again, it's not definitive, but it's the first sort of proof of concept of moving in that direction, and that's exciting. I'm getting close to the end of time, so I'll go as quickly as I can. So in the case of consolidation, I'm using consolidation here as a proxy for the ways that we might think about forgetting errors. So forgetting errors are hard to study because you can't sort of control when and what it is that someone is going to forget in general. But one of the ways that we might think about this is by thinking about the process by which memories go from being initially fragile or initially able to be forgotten into some sort of more stable and long-term form. And these processes in neuroscience and in psychology are referred to as consolidation processes. So we generally think about consolidation as happening at two levels, both molecular and systems consolidation. Molecular consolidation happens quite rapidly in the minutes to hours, maybe to days, following the formation of a memory, where systems consolidation is a much longer process. There are debates about how to distinguish between them. I'm not pursuing that here. But the particular cases that I'm talking about here are cases of molecular consolidation. And what we know from a long history of studying consolidation is that if you disrupt this process, then the result is forgetting. It's retrograde amnesia. So if I learn some information, if you teach me something, and then you come and interrupt me just afterwards, if you interrupt that process, either cellularly or personally, you can inhibit the ability to retrieve and to retain and hold on to that information. So consolidation processes are critical for holding on to information for more than a few moments. And so we know that there are standard ways in which we can use protein synthesis inhibitors, things that keep the memories from forming, that will stop an organism from remembering. So say I have a set of mice, I let them all run a maze or learn a behavior, and half of them afterwards I give this protein synthesis inhibiting solution. The other half I just give some sort of sham solution, some saline perhaps. The ones that weren't allowed, had the process of protein synthesis blocked, aren't able to remember that information. And you might pause to wonder, and actually I think historically people haven't spent a whole lot of time even wondering. What they've assumed is, oh, by failing to synthesize proteins, by failing to undergo the process of consolidation, the engram was never fully formed, or it never got past its initial fragile state, and so there was nothing there to be retrieved. So most of the time people haven't even paused to ask, when amnesia occurs in these cases, when consolidation is interrupted, is the failure with what was meant to be stored, or is the failure on the process of retrieval, or how that information could be activated? And most people just think, well, what consolidation does is sort of beef up the information that's stored, put it in a format that's more permanent. And so if you disrupt that, then you just don't have the information, you just don't have the engram. But it's been very difficult to disentangle or even think about what alternative hypotheses could be until there were optogenetics. So in this 2015 study out of the Tanagawa lab, what you see here is the mouse being able to explore a particular environment and form these light-sensitive engrams. And for the mice that are doing this, they are trained in a particular behavior. After immediately following the training, half of them are given saline, a sort of sham solution. The other half are given the anisomycin, which is the protein synthesis inhibitor. The next day they test, and you see what you expect to be are the standard results. The ones that just got saline, the ones that were so allowed to have consolidation proceed as normal, they remember the information, they remember what was involved. For the ones who had the protein synthesis inhibited, the ones that were not allowed to consolidate, they don't display the effects of training. They've completely forgotten what that information is. But because all of these mice are mice that have been optogenetically modified, the engram, the cells that were involved in the act of training them has been made light sensitive, right? It has been given that particular light tag. So even for we take the set of mice who learned the event and had that engram for what they learned becomes light sensitive, but then they're given the protein synthesis inhibitor, so they fail to consolidate that memory. They go to the next, they get put back in that context in relatively natural, at least in laboratory settings, natural retrieval situation. They can't recall the information that was given to them. But now you optogenetically activate that engram. You say, well, what happens if I turn on the blue light? And sure enough, if you expose the light, those cells turn back on and you get the behavior back. So even though consolidation was blocked, consolidation failed to occur, and you can't naturally get the organism to engage in standard retrieval behavior practices, you can still optogenetically activate that engram. And showing, in some sense, the information is still there. What consolidation has actually blocked is the means by which you would access and retrieve that information instead. And so I think I've got another page just showing you all of the various, I think this was surprising to them too, or at least that's what I read between the lines of the number of different kinds of interventions they used in order to display that, in fact, consolidation really, really was prevented in these cases, but for up to 8 days after the event in question, you could still put the animal back in the context. On their own, they won't display any memory of what happened to them, but if you turn on the blue light, you can reactivate the engram. So the information is in there. What's been lost as a result of consolidation is the ability to access that information. So consolidation, if this kind of view is right, what this suggests, in opposition to a lot of other work that's been out there on the nature of molecular consolidation, it actually might be an X4 deficit, not an engraphic one, right? So synaptic strength might not be a crucial component of the stored memory. This is a quotation from the paper. Instead, the primary role of that augmented synaptic strength during and after the consolidation may be to provide natural recall cues, right, to engage in the X4 process for the engram cells that are already there. Sort of opening up new ways of thinking about what might actually be happening in the case of consolidation, with a way of thinking about it that wasn't available prior to optogenetic intervention. And so this is a quote from Tanagawa in an interview about that paper, where he just simply says, the majority of researchers have favored this storage theory of thinking that that's what consolidation disrupts. And they actually think that this paper is pretty revolutionary in being able to intervene in ways that show that view is potentially wrong. So just to wrap up what I've said here, optogenetic technology makes it possible to make an advance on what I think is a general systematic methodological challenge for how we investigate memory, and that we care about what's stored in memory, but we only have access to what's stored in memory via what is retrieved from memory. And so that makes it very hard to disentangle the output, in the output, what was a function of the retrieval processes that were involved, and what was a function of the information that was stored to begin with. And we like to think about that distinction, but prior to the ability to intervene into engrams directly, it's been a merely conceptual one. With optogenetics, we now have the ability to do some of this intervening and to actually get in and activate the engram directly, circumventing at least standard and X4 processes. And the ability to do that makes new kinds of manipulations possible. And via having those, already within a couple of years of working within this paradigm, the Tanagawa group and others have really started to shake up a lot of the ways we've reflexively thought about our best explanations for where the disruptions are happening in cases where we get instances of false memory or forgetting or failure to consolidate or something of that sort. So I don't have a worked out view of what it takes for something to be revolutionary in science or to mark a significant development, but a tool that very, very quickly produces a set of significant results that make us look at standard processes and mechanisms different than we were before, surely is at least on the path to something of that form. So I don't know if someone can talk to me to say whether or not I'm far enough past time that I should just stop and not say these.
You can give some general lines and 3 more minutes.
Okay, that's all I'll say. So I mentioned an objection at the end of the paper, which is you might think I might have overstated my hand, that optogenetic intervention isn't actually totally circumventing the Esphory. It's actually just inventing a new artificial Esphory. And so the methodological challenge is deep. It's not going anywhere. It's a sort of more fundamental epistemological challenge for how we think about this distinction. And so, maybe I've made it better, or at least optogenetics has just made it different, but it hasn't actually made it go away. I think that's a very possible way of thinking about things. And I won't go into the details about them now, but if someone wants to talk about them later, things I've been thinking about more recently have to do with the Tanagawa group's more recent discovery, them and a few other groups as well, of something that they call silent engrams, which is this idea that there are some engrams during at least some stages of their being engrams, at least on their definition of what counts as an engram, where they can never be activated by natural or normal means, but they can be optogenetically activated. So some engrams have this sort of with what looks to be an instance of cases where their information is in there, but can't be activated by any means other than optogenetically. And I think we can ask real questions about whether those things are still going to count as engrams, what this says about whether the optogenetic manipulation is too powerful, whether it's allowing us to see things and read into them things that just aren't there. But also, I think what it's really forcing us to do as we get these more fine-grained manipulations is think about what exactly our commitments are, in thinking about the nature of the mechanism of the engram and how it will move and change and persist over time, what we think its essential and defining features are. And so I think the answer to the question about the relationship or what optogenetics has done for the neurobiology of memory is not that it solved all the problems that were out there, or even if it turns out that it does solve these problems about false memory and consolidation, it will invite as many more, and there's going to be plenty more work in that respect to be done. So I can say more about that later if people are interested, but thanks for listening. I'll stop.